अतुलं तत्र तन्तेजः सर्वदेव शरीरजम् एकस्थं तत्र भुनारि व्याप्तलोकत्रयः त्रिष्वा. That incomparable radiance that was born for all gods and pervaded the three worlds came at one place and took the form of a woman. Yatra naryastu pujyante ramante tatra devataha yatrastu na pujyante sarvastra falaha kriya. Where women are honored, divinity blossom, and wherever women are dishonored, all actions, no matter how noble it may be, remains unfruitful. Good morning, everyone. Myself, Dr. Sakshi Bharaj. On behalf of Elton Technologies, I would like to welcome you all for this Women's Day special webinar series, and thankful to all of you for sparing your valuable time. Like the last year, we have organized the event to celebrate the International Women's Day, which is celebrated on 8th March every year. The significance of Women's Day is increasing year after year and has become a custom today. It appears to celebrate respect, appreciation, love, and care towards the woman. Today, we are also celebrating 75th Republic Day. From the Altum's platform, I would like to convey my wishes to all my viewers. Happy Republic Day to all of my viewers. You know the fact that 2 days actually, for the very first time, military's Nari Shakti is at the forefront of the Republic Day parade. So hats off to all of the women out there. I feel quite fortunate actually to have been granted this opportunity for this Women's Day celebration, as it allows me to express my gratitude also to all the ladies who have contributed significantly into my life. I probably never wish verbally them, but of course, deep inside into my heart, I always thank them for being in my life and shaping it the way it is today. In my opinion, actually, women should be respected not just because they are women, but also because they are individual with their own identity. This long week celebration is especially dedicated to all the women and for the recognition of their amazing deeds. Women like Mother Teresa, Rani Lakshmibai, Sarojini Naidu, Ahilyabai Holkar, Savitribai Phule, Aruna Asif Ali, Indira Gandhi, Suchitra Kriplani, Lakshmi Sehgal, Kalpana Chawla, Nirmala Sitaraman, and the list is so long. These are such great examples of power and greatness that a woman carries actually. There is a very interesting fact behind Women's Day celebration that a woman named Clara Zetkin, who was the leader of Women's Office for the Social Democratic Party in Germany in 1910, appeals for the annual Women's Day celebration, which we today celebrated as International Women's Day actually. We at Altum think of every day as a Women's Day. However, the day itself takes place at 8th March, and it is nice reminder to all of us to keep celebrating and supporting the fabulous women around us. I would like to thank Altum for organizing this Women's Special Webinar series. Let's begin the celebration by recognizing and appreciating the female researchers throughout our webinar series who have achieved success in their respective fields actually. Today, we have such a dynamic guest speaker with us, Dr. Sonal Dubey, Professor, College of Pharmaceutical Sciences, Dayanand Sagar University, Bangalore. Ma'am has PhD in Pharmaceutical Sciences, having more than 21st year of teaching and research experience. She is active in research and as a medicinal chemist. Ma'am has 45 research and 7 review articles to her credentials. She is currently having 666 7 citations and 14 H index. She is co-author for many books chapters. Ma'am research interest is mainly lies in anti-HIV, anti-cancer, anti-malaria, anti-tubercular, molecular design, and computer-aided drug designing and synthesis part. Ma'am has expertise in the computer-aided drug designing. She has availed a cumulative grant of around 41 lakhs from the various funding agencies like DST, AICT by the Government of India, and Rajiv Gandhi University of Health Sciences by the Government of Karnataka. She has guided 16 PGs and 2 PhD students. Ma'am also worked as editor for many journals, basically. Along with being on the board of various other journals, she is also on a reviewer board of many journals like Elsewhere, Bentham, and Spingers. We are honored to have you, ma'am, today with us.
Thank you,
Welcome you,
Sakshi.
ma'am.
Thank you, Dr. Sakshi.
I request all my viewers to stay connected with us throughout the session and feel free to drop any queries into the chat box. We will be taking the question and answer at the end of the sessions. So, without any further ado, let's start the session. Up to you, ma'am.
Thank you, Dr. Sakshi, so much for such an elaborate introduction. Uh, Let me begin with my presentation. Am I audible?
Yes, ma'am.
No. Okay, so at the outset, I would like to thank Alchem Technologies for arranging this kind of a workshop. Not a workshop, like a seminar series, which you people have arranged over the period of time till the Women's Day. So it's a very good effort from your end to recognize the women which are doing good in STEM. I would thank Alchem for inviting me for one such lecture today here. So today I'm here to present a talk on published research which was published in uh, 2018, and it's titled as the study of pyrazolo-benzimidazole derivative as spleen tyrosine kinase inhibitors and in-silico approach. So I'll tell about this, who has done the work and all towards the end of the session. So we will begin with the presentation right now. Before I begin, as Dr. Sakshi correctly told that it's a Republic Day today. It's our 75th Republic Day. So I would like to wish all our participants a very happy Republic Day. And as Prime Minister Modi has given us, along with Jai Jawan, Jai Kisan, he has included Jai Anusandhan also in the logo, which is a very apt thing right now because India is growing very well scientifically too. Other than economical development, they are growing. We have seen Chandrayaan, we have seen Aditya, Gaganyaan is on the way. So Anusandhan is going in a good direction right now in India. And as women, we would be equal contributors. Almost 50% of the population, we should be there in the 50% of the research which is happening all around India. So with this, Happy Republic Day. And as the theme of this seminar series is Women's Day. So International Women's Day 2024 is themed as Inspire Inclusion. So which Altum Technology is doing very rightly. They are inspiring the inclusion by there are so many ways in which you can inspire the inclusion, like recruiting, retraining, developing female talent, supporting women and girls into leadership, decision making, business, and STEM, and providing women the equal opportunity for education. So I congratulate Eltem Technologies for taking up the initiative and inspire the inclusion and going ahead with the theme of this year's International Women's Day. So coming back to the presentation, the study of pyrazole-benzimidazole derivative as spleen kinase inhibitors and in-silico approach. So spleen tyrosine kinase is a member of cytoplasmic protein tyrokinase family. The work of this family of enzyme is it is having a reciprocal connection between the mechanistic pathway and the inflammatory factor. So if we are going to inhibit these enzymes, the protein tyrosine kinase group of enzyme, we have to pick the ones which we want to target based on our study. We are going to, if we target any of these enzymes, we will be inhibiting an inflammatory pathway. So we are interested in autoimmune diseases and hematological tumors are the diseases of our interest for this study which we had taken. So we have taken out of the protein tyrosine kinase family, spleen tyrosine kinase, and tried to develop certain inhibitors, uh, computer aided using computer aided drug design to come out with certain say models which is going to give you a good structure in pyrazolo-benzimidazole group of series. Okay, so that was our target. That what work we have done, we want to have pyrazolo-benzimidazole type of derivatives. We want to understand what are the different features which we should have in it so that we can find a good lead in the series. So this is a structure sequence of the SYK kinase. SYK is spleen tyrosine kinase. So SYK structure sequence is like this. You have an NS2, NSH2 interdomain A, CSH2 and interdomain B. So this NSH2 and CSH2 is separated by an interdomain A, and on the other side we have interdomain B. If the organizers can tell me that if my cursor is visible to you people, is it
No, ma'am.
visible to you people? No. But my slide changes are happening, right?
Yes, yes, ma'am. It's happening.
Okay. Okay, then without cursor only, I'll try to explain them. So this NSH2 and CSH2 domains, in between this interdomain A and interdomain B, there is a phosphorylation process which occurs which causes the activation of this protein. So as such, the protein as such without phosphorylation is in its inactive form, and when the phosphorylation occurs, it becomes an active compound. So this is the different receptor signaling for the hemopoietic cells, the two diseases, autoimmune and hemopoietic cells, which we targeted. So these are the different types of the pathway which SYK is influencing. Now, if you look at this particular picture, the top one is a ribbon structure and the bottom one is a solid structure. What I wish to make you people understand in this, look at the solid structure where it is written active. Okay, and this side it is the B side of the figure. The left side it is written inactive and the right side it is written active. So this SYK itself, when it is not phosphorylated, is in inactive form. When it is going to be active, it has to get phosphorylated, and you see this active site shape changes and it becomes active. Okay. So this is the structure of the active protein which we have. This is what the right side of this yellow zone, if you can see in the figure B, yellow zone of the protein, you will see that this is the shape where the active is written, is the part, the way it is required to be active in the active site, the where the binding will happen with your ligand. Okay, so the enzyme has to convert itself into from inactive to the active form before the action can be exerted. Now, we have done a lot of literature survey and we found many heterocyclic compounds which are active in that particular SYK as an SYK inhibitors. So these are the structures of some of the very well known compounds which are there as SYK kinase inhibitors. Okay, so as you can see, all of them are heterocyclic molecule having some or the other kind of like a benzimidazole type of structures in between them. So there are other categories as well, but we became interested in the benzimidazole type of a compound because that is the one we have taken up for our study. So pyrazolo-benzimidazole kind of derivatives we have taken. What we did is, sorry, what we did is we are using the computer aided drug design to come up with the a particular model, a particular pharmacophore, and to let us know where all the binding is happening. So we are going to use this computer-aided drug designing for all these purposes. For that, what we had done is, first we have done the QSAR, followed by pharmacophore modeling, followed by molecular docking. Okay, so for this particular study, we had taken 55 molecules containing pyrazolo-benzimidazole scaffold with a varied IC50 ranges in it, which is a requirement to make a good group. So first one out of the 55 compounds, we have done the QSAR studies. In the QSAR studies, we have taken training set and test set molecules. So we have divided the 55 compounds into 38 of the training set and 17 as the test set compound. So why we have to divide our compounds into the training set and test set? This is for the benefit of the young researchers who are interested in QSAR, but they have not done it as as much as they would have wanted to do it. I would like to explain here that when you do a quantitative structure activity model generation, you need to make a model which will give you the results you are looking for. Like you want to, what does a QSAR do is, it is going to give you a model and you are going to use that model to predict the activity of the future compounds which you will design first in your mind, and if it is showing activity, you're going to design it, make it in the lab. Okay, so that is the purpose of a QSAR model. But if you're generating a model, it should be a good, valid model. That the model is actually worth that we can use this model to generate a new, to generate the active or inactive, to let us know what is the active or inactive part of the and active or inactive molecule. So that is the purpose of a QSAR model. So when we do a QSAR modeling, we divide our compounds into training set and test set. What does the test set do? Test set will help you, training set will help you to generate the model. Okay, so you have used certain number of compounds to train or to make the model which is going to predict the future compounds. But whether we have made a good model or not, we are going to have some test set molecule, which is for the validation of the model. So what does the test set do? You give your test set molecules to the model and see whether the predictive ability of the model is good or not. So if the predictive ability of the model is good, that because for the test set molecules, what are the test set molecules? These are the molecules of which you know the structure, of which you know the activity as well. So the activity part you will not give to the model. You will ask the model to predict the activity. And then you compare that predicted activity with the actual activity which you have found in the lab. If those two things match, that is, the predicted activity and the actual activity match, means the model which you have generated using the training set molecules is a good model. What does a good model mean? Good model means that we can use this model for predicting the activity of the design compound in the series. Okay, so this is the basic background of a QSAR. That how what do we do with the QSAR model when we generate it? But there is a prerequisite for the QSAR modeling, that you must have a data set which has similar kind of structure scaffold. Like the basic nucleus should be same with the variation in the substitutions. Okay, so that is the basic requirement for the QSAR model. So whenever we are doing QSAR studies, we must have a series of compounds with us with the same basic nucleus in them. In this present case of the study, what we are doing pyrazolo-benzimidazole derivative. So that is our basic nucleus. So we have collected from literature 55 such compounds and divided them into training set and test set. You must keep in mind whenever we are dividing our compounds into training set and test set, your training set should contain all types of compounds. That means it should not only have the active compounds or inactive compounds. It should be a mixture of both, a good mixture of both. Okay, active should be more, but there should be inactive compounds as well. Same is true for the test set molecules. Whenever you are having the test, making the test set molecule, you're keeping certain molecules aside from your library of the database. So those molecules should also be mixed in nature, active, inactive together. Okay, so that we are going to test the ability of the model to predict good as well as not so good molecules. The model should be able to inform us this thing. That is the purpose of generating a QSR model. Okay. So with this basic in mind, we have done certain QSR studies, QSR model generation and validation. Validation is when you give the test set to the model and ask them to predict the activity, and if they predict it good, it is a validated model. If the prediction doesn't go well, that means your model is not okay. You need to change certain parameters which you have taken in the model, or change or add certain more library data into it, more structures into it, more variety into it, and regenerate the model. Okay, so any QSR model which we develop will not become a good model. The validation will prove whether it is a good model or it is not a good model. Okay, so we have taken 38 molecules as the training set and 17 molecule as the test set molecule for QSAR modeling. So we have taken both actives and inactives, and we have done a partial least square method to do this. So that is a statistical parameter. I hope many of you understand partial least square method. And whenever we do PLS, we have to keep certain factors. So we keep on increasing the factors, say 2 factor, 3 factor, 4 factor, 5 factor, 6 factor, like that. And we keep on generating the models. And we see with changing the PLS factor, is the model becoming better or not? So till the time the model is becoming better, we can keep on increasing the factor in the PLS. But if the increasing the PLS factor is not giving a drastic change in the model, we will not change the factor further after that. Okay, so for this study, what we observed that PLS factor, when it was kept till 4, we got a good optimum modeling. Increasing the PLS after that was not giving a good, any significant better result, significantly better result. So we kept the PLS factor as 4. Okay. So the structure feature which we constructed the model, the test. So when we call validation, there are two types of validation. There's an internal validation and there's an external validation for the model which you have made. So internal validation is that from the library only, like what we have done, segregated the 55 compounds into 2 batches of test set and training set. That is an internal validation when we do. Okay, external validation is from some other place you are getting it and then you are validating your results. So the technique which we had adopted was internal validation. And whenever we are doing QSR, the activity range should be good. Okay, so good activity range means you should not only have an active molecule and inactive molecule. We need to have log IC50 value, and those values should vary between like 3 log values should be there. So the variation in the activity range should be there. It should not be like a little active, inactive, little more active, inactive. You should have a variation of 3 log activity in the structures which you are segregating for QSR studies. And when you have to do the QSR, you have to draw the structure, you have to energy minimize it. Okay, so you do the energy minimization, you do the optimization, you do the conformation generation, because each molecule will have its own variety of conformation in which it can exist. Then energy optimization will depend on the conformation and the configuration of the molecule which you are taking. So we have to take into consideration all those things when we are doing QSAR studies. So we will do this all and we will generate a model. Now this is the figure which is telling you that whether our model which we generated and which we took finally, how much is the robustness of that model. So as you can see, you have predicted activity versus the actual activity graph. So for both training set as well as test set, we asked it to predict, and we got a pretty good R square value for this. That means our predictive ability of the model is the closer these points are to each other and to the center median, that R square median, that means the predictive ability of the model is good. Okay, so this is how we come to know that our model will give you a result which is actually going to be right. So these are the parameters which we had taken, which we got for our QSAR model. So the few things which we should keep here in mind and look at what is good here is R square value, which is of importance, and Q square value, the cross-validated R square value. Okay, so whenever you look at a QSR model, these are the two things you're going to look at first, and the other things you'll later. So if you have a R square of zero point 0.8 and above, and Q square of zero point 0.6 and above, we consider that model as a good model. Okay. So these are the things which we got in the model. As you can see in the result, we got R square value and a Q square value of the desired values. So we took these model and went further in silico studies. The next thing we did with these molecules was pharmacophore modeling. So let us have a look at what is a pharmacophore modeling. Pharmacophore is an abstract description of molecular features that are necessary for a molecular recognition of a ligand by a biological molecule. As per IUPAC, pharmacophore is defined as an ensemble of steric electronic feature that is necessary to ensure optimal supramolecular interaction with a specific biological target and to trigger or block its biological response. So, what does this mean? This means that when we are doing a pharmacophore modeling or we are generating a pharmacophore, means we are going to take out the features which are electronic and steric in nature, which are required for that particular activity. Let it be an agonist activity, let it be an antagonistic activity. Okay, so what are the features you require in a molecule which you are going to design, say, as an antagonist for an enzyme, where in the molecule you will like to have hydrophilic group, where you will like to have hydrophobic group, where should be the electronic groups. Okay, all those placements of the groups, when it tells you, it becomes a pharmacophore. Okay. So a pharmacophore explains how structurally diverse ligands can bind to a common receptor site, and a pharmacophore model can be used to identify through a de novo design or a virtual screening novel ligands that can bind to the same receptor. Now, what is the difference between a QSAR study and a pharmacophore modeling? As I've told you earlier, in the QSAR studies, we must have our library which has a same basic nucleus. Okay, but in real life, what happens? A drug which is like you have a target enzyme, you want to inhibit that enzyme, but the type of molecules which are binding to the enzyme may not have the same basic nucleus. Okay, so there will be a diversification of the molecules which are going and binding to that particular protein. In that case, how do we do QSAR study? We can't do QSAR studies, right? Because for the QSAR, the mandatory thing is you should have structurally, like a basic nucleus should be same. Now, if you have varied structure, but they are all showing same kind of inhibitory activity, how can we use these structure to come out with something which will tell that this kind of, if you have a steric group here, if you have an electronegative group here, and electropositive group there, is going to give you a possibly active compound. That thing can be done using pharmacophore modeling. So that is the basic difference between QSAR and pharmacophore modeling is QSAR requires a basic nucleus. All the libraries should have same basic nucleus. Whereas when you're doing pharmacophore modeling, you may have or you may not have. You can do using both. You can use using structurally diverse compounds also. You can do a pharmacophore modeling. Okay, so that is the basic concept of the pharmacophore modeling. So there are many pharmacophore modeling tools are available. I have named a few. Okay, you can choose depending on basically on your budget, how much you can invest, and what is the robustness of these. You can go to the different research libraries where you find the literature and what kinds are giving the actual result, what kind of are better giving you the better kind of models which are having good predictability. So these are all the different kind of softwares which I have invested here. So pharmacophore pattern, what it will tell you about the hydrogen bond donors, hydrogen bond acceptors, positive features, negative features, aromatic ring, and hydrophobic feature, and their combination. So, what are these things which are required in your particular case of interest, your enzyme of interest, where you require these features? Okay, these are the features which a pharmacophore pattern will tell you about. So, looking at the pharmacophore pattern, you will come to know where you need to put a hydrogen bond donor in your structure, where you need to put a negative feature, aromatic ring, or a hydrophobic feature. So, all these things you will come to understand from the pharmacophore model. So, it includes several patterns arranged in a particular three-dimensional way. Each pattern is depicted by a typical sphere containing radius that determines the deviation tolerance from the exact position. That means how much you can deviate from the position it has given. This is an example of a pharmacophore model which we have generated for our study. So, as you can see, these rings are the places where it is telling that you need to have these kind of groups here. Okay, so this is a overlay of the active molecule which has generated a 5-point pharmacophore hypothesis. So you can say it a pharmacophore model or a pharmaco hypothesis, whatever you want. Basically, it is a pharmacophore generation. Okay, the different softwares will give you the different terms. Okay, the terms can be anything, but basically it is going to give you where and all you need what and all type of these things, which I have explained here. Hydrogen bond acceptor, donor, positive, negative, aromatic. So basically these features are going to tell you about this. So we have generated these two kind of a model by overlaying. These are the overlay of the active compounds and this is the overlay of the inactive compound. So what did the active compound contains? Where in all they had the hydrogen bond donor, where in all they had the acceptor, and where in all the inactive compounds had these types of features. So if we want to make something good, we should know what is making it bad, right? Then only we can avoid the bad things and only approach for the good things. So that's why we have generated an active molecule pharmacophore and an inactive molecule pharmacophore. To understand the difference between two, that which features are making the molecule active and which features are making the molecule inactive. So what we did, we have generated many pharmacophore models. These are the names which were given to the in the table under the hypothesis, the names which are written there. Since I cannot use my cursor, so under the table hypothesis, these are the different names which were given. Out of these models, we have selected the first one, which is ADRRR underscore 2, which was which was giving us the best possible result. So how do we check that? Again, we check depending on the different scores which we get, different statistical parameters which we get when we make the pharmacophore, and based on that we decide that which one we have to opt for. Okay, so out of all these 1, 2, 3, they are ordered in the order of the relevance, the predictive, like the one which one is the best, the second best, and the third best. So we have taken the first top one. It's a 5 point pharmacophore model. So it has one hydrogen bond acceptor, one hydrogen bond donor, three aromatic rings were there, which came out from the software that these are the essential features. If we want to design certain molecule which will be SYK kinase inhibitor, these are the things which you require in the molecule to be and at that particular places. Okay, so if you can see the distances between these two and these two, the software will tell you, and how much of the deviations you can do, all those things you can come to know. So we can have the best possible model depending on the statistical parameters which will come with the model. Whichever software you're going to use, you will come out with certain statistical parameter which will tell whether the model is good or not good. So we have to do this kind of we did this kind of a study and we came out with AD underscore 2, which was the best model for our particular study. Okay, so now you see this figures. I hope you are able to see this blue color cubes which are here and the red color cubes which are there in the other figure. So this is a representation of the a model which is giving you where the hydrogen bond. Now basically I'm coming to the overlay of a pharmacophore modeling and the QSAR. How we have used the QSAR and the pharmacophore modeling to understand the features which are required. Okay. So as you can see in the first figure, which is on the left hand side, you have a model which is giving you the same pharmacophore feature which is come AD underscore 2. The same kind of model has come and it is giving you the these blue cubes are telling you from the QSAR model where we have the hydrogen bond donors are required. So these are the molecules from the QSAR studies and the 3D and the pharmacophore. Both the results are depicted together right now. Okay, so the same molecules, because we have taken the same 55 molecule set, which we have done the QSR as well as the pharmacophore studies, and we try to find out where both the things match, which are the compounds in which the QSR model as well as the pharmacophore model are being matched. That means we are going to filter our compounds. We are not going to take all 55 compounds for further studies. We are going to sieve them and take only the active ones with us for our next step of the study, which is molecular docking studies. Okay, so we have found out from this overlay that these are the places where the blue cubes are there. These are the places where you have the hydrogen bond donors in the compound. So this is the place if you put, so if you're going to have a 5 point pharmacophore hypothesis, you are going to, like as I explained earlier, there are three aromatic rings, there is a hydrogen bond and all those things. You need to have a hydrogen bond donor at certain place, which is depicted by the blue cubes. Okay. So similarly, we have other features like hydrophobic effect. So this red and blue again cubes you will see. These are the places where the hydrophobic effects are there, and these are for the inactive molecules. So active molecule and inactive molecules, pharmacophore hypothesis we have seen, pharmacophore model as well as we have seen where the QSAN and both are merging and filtering our compounds. Similarly, we have seen the electron withdrawing effects where and all it is necessary. Okay, so you will have to look at your compound structure, look at these things and filter your compounds. So after doing all this, we came out with 7 best compounds which were giving us good results in QSAR as well as which were fitting very well in our pharmacophore model. Both the things were good in it. So what we did, we have taken those 7 compounds and we have done the molecular docking studies of those compounds. Now, molecular docking studies, as you might be aware, that we have to take a structure of the protein, we have to take our ligand, and we have to try to fit our ligand into the active site of the protein. Okay, so there are so many steps which are involved. There are different softwares which are there. There are certain ones which are free in nature. You can have, if you have money to buy, please go ahead and buy the ones which you want. So you can do with anything whatever you want to do, depending on basically your like which software you like and how much budget you have. So you can use, and also how much is the consistency of those softwares which you're using, whether they are there valued in the research field as a good software or not. Every other software, please don't go and use. See the research, do the research properly, see the literature properly, and see the ones which are more acceptable. Because ultimately, when you're going to do the work, you need it to be published. Okay, so please see to it that you're choosing the right one, which is going to give you some result which is more acceptable in the literary field, in the scientific literary field. Okay, so for our compounds, we have taken this protein, 3V-S8 protein, from the protein data bank. So this is a protein data bank. I'm sure all of you know that it is a place where you can get the protein from, and then we have taken our molecules and we have done the docking studies. So these are the results of the docking studies. We have taken 2 standard compounds, 5-fluorouracil and nocodoxazole, to compare our compounds with. As you can see, the binding energies of the standard compounds were not as good as the ones which we have taken for the studies. These are the results of the docking pose. So you can see that I think the figure on the right side is more explanatory. You can see that which part of the molecule is binding to which side of the which amino acids, which sequence it is binding. So you're going to get the results according. You come to know that these are the amino acids which are involved in the docking studies. These are the which are when the interaction is happening actually with the protein and the ligand. These are the amino acids which are there actually actively participating in the binding. So these are the amino acids which we need to target to bind. So with our study, what we have come out with that all the active compounds, they have a good hydrogen bonding with these amino acids. So this is the sequence, the number in front of the amino acid is the sequence of the amino acid in that particular active site of the protein, like med448, leucine 377, leucine 306. So like that, we got all the information about the amino acids which are binding with our ligand. Okay, so this docking studies is going to tell you all about these kind of studies. So what we found out was that we need a hydrophobic group and a hydrogen bond donor, which are interacting with the leucine 377 and glycine 378. And aromatic hydrogen bonds were also seen with alanine and leucine at different amino acid sequence at 451 and 502. And there are other aromatic rings which are attached to pyrazolo scaffold, also showed aromatic ring interaction with glue 449. Okay, so these were the interpretations of the studies which we had done. So in conclusion of our study, what we had come to understand is that using 3D QSAR, pharmacophore generation and docking studies, we were able to find out that a hydrogen bond donor, a hydrogen bond acceptor, and 3 aromatic rings are essential to design a novel potent SYK inhibitor. kinase inhibitor. Okay, so if we are going to keep these things and we are going to use our 3D QSR model places where it has shown the hydrophobic interaction, the hydrophilic interaction, the electrostatic interactions, and steric factors it has shown. So if we take clue from the pharmacophore model, from the 3D QSAR model, and we know from the docking studies which amino acids we are going to target, we will, there is a very high chance that we are going to design a good active molecule. Okay. Please remember, in the end of any computer aided drug designing work, it is just a probability. Okay, there is no 100% guarantee that you are going to get an active molecule. But by doing all this, what we achieve is a probability of getting an active compound more than rather than just going by a hunch that I think I should do this. Let me fix this NH2 group at position 4 and let me put a hydroxyl group at position 8 and let me see what is the compound's activity. So I am not doing anything based on hunch. If I'm changing a molecule's atoms or a functional groups or anything, I have a rationale behind it. I have a logic behind it. I am supported by certain statistical parameters. So computer aided drug design is a very good tool to help you decide doing that. Okay, but there are no guarantees in computer aided drug design, but the probability of getting becomes multiple folds more than just doing it by a hunch. Ultimately, you will have to go to the lab, design, synthesize the molecule, and test it in the lab. Okay, but your rationality increase multiple folds, your probability of getting an active compound increases multiple fold. So I would urge all the young people here who are working in this field, before going for the lab synthesis, please always go for a computer aided drug design for any interest, any target of your interest. Okay, whichever you're working on. So this is a good tool. Computer aided drug design is a tool which we have, which we should use it to our advantage. Okay, so with this, I'll conclude my talk about this particular study of pyrazolo-benzimidazole derivative as SYK inhibitors, SYK inhibitors. I would like to thank RGHS Bangalore for providing financial assistance in the form of a research grant, with the help of which we could complete this work. And I would like to thank my co-authors in this paper, Dr. Pravika, Dr. Sakshi, and Dr. Rekha, who had been instrumental in doing the research work. And on this day, I'm very happy to say that I chose this particular paper to be presented today because since Alchem Technology is doing this as a Women's Day, it's a beginning and we are doing a celebration of Women's Day over a 3 month period. This paper is all women paper. Okay, so I wanted to present this work only for this particular talk for the day, and this is it has been published in Structural Chemistry in Springer Journal. It is a Springer Journal. So I thank my co-authors as well for their contributions to this particular research. Thank you, everyone. And I came to know from Net that this is the pose for the year for Inspiring Inclusion pose. So thank you, everyone.
Yeah, thank you so much, ma'am. It was very well explained, I would say. section, and it was very informative. You have explained very clearly about the QSAR and the pharmacophore models, how to generate them, and what is the basic difference in between the QSAR and the pharmacophore generation. Because I have seen many students who having this confusion in between the QSAR models and the pharmacophore generation. Why we are doing basically those two models, and how could they help us in doing further while taking those models further for the docking studies and the further for the simulations, how they useful to us, how important they are having by generating those QSR and the pharmacophoric model. Of course, we have few questions, ma'am. Let me take those questions. First one is, can we predict the pharmacophore of the compounds without biological activities also? You're on mute, ma'am.
I need to
Give
give some rights. Yeah,
Yes, yes, ma'am. You are audible now.
audible now.
Yes, ma'am.
Okay. So you cannot generate any model without activity. Without activity, how will you know which are the feature here which will give you a good compound or good results? So you need to have activity. Without activity, it's not possible.
Adding to you, ma'am, actually, in Discovery Studio, we have two, three algorithms. Like you have mentioned, the catalyst, hip hop, and the hypogen algorithms. So we have two categories. If you are having the components with the activity, if without activity, so you can go with the qualitative structure-based pharmacophore generation, where you can align two active molecules to generate their pharmacophoric features, and you can have that model further for your studies. So yes, we can have that option. We have that option in the Discovery Studio. If you want to explore, I can help you out there. Yes. So can we have the second question? Okay. How to identify the binding site of an apoprotein? Actually,
For an apoprotein, that is a little tricky because apoprotein you will have the it's not the usual active site which you will get, but if you can get a PDB structure which is binded to the apoprotein site or something, then it is you can do it. But I don't know about Discovery Studio. They have a software which
yes, yes, ma'am.
works like that.
Yes, ma'am.
I think
Actually,
Dr.
in
Sakshi can help.
Yes, ma'am. In Discovery Studio, we have three different tools to identify the biological activity. Actually, apoproteins means when you don't have any inhibitor into it, right? So whenever we are downloading the PDB structure of your any protein from the databases, you have first requirement is you must have the inhibitor. But if not, then you can identify the binding site on the basis of we have two, three algorithm, like we have the flood filling algorithm there, we have the eraser algorithm there, which would help us to identify the binding site even in the apoprotein itself. So yes, we can identify the binding site of apoprotein also. Yes.
So I guess
Next.
BioVia Discovery Studio will help you with this.
Yes, definitely. Next question, can we have next question? Okay, so QSCR models or 3D QSCR model, which is the best for the drug designing?
QSAR model can be a 2D, it can be a 3D, it can be 4, 5. Nowadays, even people are using 6 and 7 dimensions in QSAR. Okay, so QSAR model and 3D QSAR are not different. 3D is a part of a QSAR model. So in three-dimensional QSAR, what do? We see the structures in a 3 dimension. In 2D QSAR, we see them like how we draw it on a paper. Okay, so if we draw it on a paper and we look only at the topological features like the bond distance, bond angle, only these things we are seeing, then it becomes a 2D. But when we see the depth as well, and you see so many more features in the structure, and you use all those features, all those descriptors you use to make the QSA model, it becomes a 3D QSA.
Yes. Yes, ma'am. Next question. Which hydrogen bond acceptors and donors can be used?
It depends on what you are looking at, what is your basic structure. But like groups like NH2, OH, okay, those which you can have the hydrogen bond with. So you need to have an electronegative atom which can bind with the hydrogen, or you have an hydrogen which will bind to the electronegative. So it depends on where you're looking and which part of the molecule you're looking at. Does your molecule contain a heterocyclic atom, or is it containing the the hydrogen in it, and you want to bind it with something with the amino acid? So it depends which part of the, whether you're talking about the ligand or you're seeing the binding with the amino acids which are there. So amino acid structures you will know, and you will see, because you know which amino acids are binding to the ligand. So what you will do is you will see the structure of the amino acids, see the structure of the ligand, and see where the electronegative or heterocyclic groups are there, and then try to bind with the hydrogen, or vice versa.
Yes. Uh, do we have any other questions? Do we have any other questions? Kindly put into the chat box.
We have finished all the questions, ma'am. There is nothing left, ma'am.
Okay. Okay, thank you so much, ma'am, once again for answering all the questions and for your great presentations. Hope my
Thank you.
viewers have
Thank
definitely
you, Dr.
get
Sakshi.
something. Yes, it was
It
pleasure
was
to
a
have you.
privilege
Yes,
to come here and talk to you, all of you. And thank you, Altum, for providing this platform to all the women which you are doing right now as a part of the International Women Day. So I congratulate you for this particular project, and thank you so much for inviting me and making a part of this.
thank you so
Thank you.
much, ma'am. Thank you, all of you.